So, Sylvia, what exactly are microscopes?
Microscopes are tools used by scientists to help magnify objects that they want to study. You can use them to look at almost anything that is too small to see with the naked eye. It's kind of like a way for scientists to explore an entirely different world. There's so many reasons scientists use microscopes, like to look at the structure and size of cells and their organelles, see how cells move, figure out where proteins are located in cells and tissues, diagnose diseases, see what happens to cells and tissues when you alter genes, plus like a million others.
Wow, microscopes can do a lot. But why are there so many different types of microscopes? I mean, you have light microscopes, and confocal microscopes, and transmission electron microscopes, and scanning electron microscopes, and atomic force microscopes, and dissecting microscopes, and...
Don't forget super resolution microscopes. To understand why there are so many types of microscopes, first you have to learn about resolution. Resolution is basically how close you can place two objects and still tell that they are separate objects. Each microscope has a different resolution, so each microscope can look at different size specimens. To explain concepts like resolution, scientists like to use math.
Oh, that sounds hard.
It's not so hard, Annabelle. Let me explain. The way scientists define the resolution of a microscope is with something called Abbe's equation. Abbe's equation is used to calculate the size of the smallest object that can be resolved by a microscope. The microscopes in this center use one of two different sources of illumination, photons or electrons. Light microscopes use photons as the source of illumination. Photons are little packets of light that travel up and down like the waves of the ocean. The distance from peak to peak of the wave is known as the wavelength. Electron microscopes use electrons to illuminate a sample. Just like photons, electrons also travel like a wave, but the wavelength of the electron is way smaller than the wavelength of the photon. Abbe, the really smart scientist whose equation we just looked at, discovered that the resolution of a microscope is limited by the wavelength of the illumination used to form an image. In Abbe's equation, the wavelength is shown by the symbol lambda. The bigger the lambda, or wavelength, the poorer the resolution. The smaller the lambda, the better the resolution. Now, since the wavelength of electrons is way smaller than the wavelength of photons, this means that...
I know. That means that electron microscopes have a better resolution than light microscopes.
You got it, Annabelle. See, math isn't so hard. Now, to give you some perspective on resolution, let's compare the resolution of the human eye, a light microscope, and an electron microscope. With the human eye, objects must be at least 0.2 millimeters apart to tell that they are separate. Light microscopes have a better resolution than our eye, and they let you see things as small as 0.2 microns. That's 1,000 times smaller than what you can see with your eye. But the electron microscope has one of the best resolutions, at a whopping 0.2 nanometers. That means you can see things 1 million times smaller than the smallest thing you can see with your eye.
Whoa, that is amazing!
Now you hopefully understand why there are so many different types of microscopes. Scientists have to use the right one depending on how small of an object they want to study. So for things like cells and tissues, scientists can use a light microscope, but for smaller structures like viruses and proteins, they need an electron microscope. So now that you've learned about why we use different kinds of microscopes, let's go see them.